Computer exercise. Design of PCR and PCR-RFLP experiments. This presentation will show how to use the online exercises on the In Silico website. The service is totally free, and we encourage students and instructors to use it as a companion to their molecular biology studies. The computer exercise may be an easy-to-perform mechanical exercise, or a complete process used to learn many molecular biology concepts, such as the relationship between DNA and protein sequences, the concept of alignment, how primers are designed, what stringency is in a PCR reaction, what restriction endonucleases are, their types, and how they work, and the concepts of typing and clustering. It is the responsibility of the instructor to make this exercise useful for their students. To start using the service, the user must select a username. Two exercise options are available: a short exercise which requires designing a PCR experiment, and a longer one which requires designing both PCR and PCR-RFLP experiments. This presentation will describe how to complete the long exercise. After selecting an exercise, click the button to enter the service. The service will generate a problem in each session for each user. In every step of the exercise, users will be shown the problem, the steps the user must overcome, and the questions to be solved in each step. The problem requests the user to design primers for a specific gene of E. coli K12 strain. In each session, 1 out of 750 genes will be randomly selected. Once the primers are designed, the user must check whether those primers are able to generate PCR bands in 2 different E. coli strains, E. coli K12 strain and a uropathogenic E. coli. If the amplicons obtained during PCR are not identical, a PCR-RFLP may be designed so that a restriction endonuclease must be selected to finish the experiment. At the end of the exercise, the service allows one to check PCR and PCR-RFLP results with the selected primers and enzymes against additional E. coli strains. The first step of the exercise is to retrieve the sequence of the assigned gene. The service will ask the users to paste the DNA sequence of the gene into the form. The sequences from strain K12 and the uropathogenic strain are requested. The users may search for the sequences at NCBI or other sources, or within our server by clicking on the ORF search engine link. A new window will open, and a form will allow the user to find the requested sequences. In the form, the name of the gene and the genome of the corresponding strain must be selected. In this exercise, only sequences from the chromosome of E. coli are requested, so it is not necessary to search the plasmids. In fact, no plasmids were described for E. coli K12 and the CFD0053 strain. Finally, the data must be submitted, and the service will show the matching genes. When only one match is obtained, as in the example, information about the gene is immediately shown. The start and end position of the gene in the bacterial chromosome as well as its length, the sequence of the gene in FASTA format, and finally the open reading frames, or ORFs, the sequence belongs to. To complete the first step of the exercise, the sequence of the gene must be copied, and the sequences must be pasted in the form, the one from E. coli K12 and the one from the uropathogenic strain. Then, the form is submitted to check the sequences. If the response is correct, clicking the next link will allow the user to go to Step 2. In Step 2, the user must find out whether the DNA sequences of the selected genes are identical and whether their protein sequences are identical. To answer both questions, the DNA and protein sequences must be aligned, and the number of nucleotide differences and the number of peptide differences must be computed. The service uses an open source program called ClustalW to align the sequences. First, DNA alignment is performed. In this case, the service will show the typical clustal alignment for DNA. The alignment will show 50 bases of the genes per line. When a specific position of the nucleotide of both sequences is the same, as for example for the first 50 bases, then asterisks under each pair of bases will indicate the match. When nucleotides in both sequences are not identical, as for example in the 8 positions shown in the image, the asterisks are absent. To facilitate the counting of differences between sequences, the service shows the number of differences in the bottom of the alignment. In this example, the number of differences to be recorded is 8. Next, protein alignment is performed. In this case, the peptidic sequences are shown. As in DNA alignment, asterisks indicate matches for amino acids, and absence of asterisks indicates the existence of differences. Again, the service shows the number of differences at the bottom. In this example, the number of peptidic differences to be recorded is 4. Finally, the recorded data may be used to fill the form. In most cases, the number of nucleotide differences is higher than the number of peptidic differences. It may be important for the instructor to point out the reason. To finish this step, the answers are checked, and in this case, the answer is correct. So we will go ahead to the next step. In step 3, primers for PCR must be selected. Again, an open source bioinformatic tool is used to pick primers, the program Primer3. To design primers, the service uses default configuration. Primers may be computed from the E. coli K12 strain, or from the uropathogenic strain. In this example, primers obtained from the E. coli K12 gene will be requested. The program Primer3 will suggest up to 5 pairs of primers. For each pair of primers, the program provides the following information. The left primer, also called the forward primer, is described on one line, and on the next line the right primer, the reverse, is described. Both primers' start position within the sequence, as well as their lengths, are shown, as is the GC content of each primer and the melting temperature, which are closely related. Additional information is also provided. The simulation program adds a button just below each pair of primers for easier selection of primers. For the first pair of primers yielded by Primer3, which is also the first recommendation of the program, the complete DNA sequence, and the position of both primers within the sequence, the forward primer and the reverse primer, are shown. Amplification will generate copies of the sequence between both primers. For the remaining 4 pairs of primers, only the basic information is provided. All it takes to select the pair of primers is a click on one of the buttons. In this case, the first pair of primers will be selected. The program will show the selected primers. This selection may be modified by repeating the previous step if required. If a new selection is not necessary, the user may skip to the next step. Step 4 requires the user to select the reagents for the PCR reaction. The service shows 12 components. The order of the components is randomized each time they are shown, and the user must choose 6 of the components. The instructor should explain to students the reason that some reagents may be chosen but not others. To select the reagents, click the button. If the selection is correct, the selected reagents will be shown, and the user will be allowed to go to the next step. Step 5 simulates the PCR experiment. the selected primers, and the genomes used in the experiment are shown. The service allows for modification of the stringency of the PCR reaction by allowing mismatches between primers and target DNA. Up to 2 mismatches are allowed by the simulation program, and mismatches may be limited in the 3' end. It is suggested that the instructor point out the importance of those limits and their relationship with stringency and PCR amplification. Finally, the maximum length of the amplified bands may be modified. In this example, the PCR amplification will be performed with default stringency values, that is, without allowing mismatches. The PCR results are shown as text, and also shown as a simulation of the band pattern after electrophoresis in agarose gel. This experiment, which did not allow mismatches between primers and target DNA, obtained amplification of E. coli K12. The service shows the length of the amplicons and their positions within the genome. By clicking the position, the sequence of the amplicon may be retrieved. But in this experiment no bands were obtained for the uropathogenic strain. The lack of amplification is also observed in the electrophoresis. In order to try amplifying the band in the second genome, the user must go back to the form and modify the stringency of the experiment. The stringency will be higher when increasing the number of mismatches allowed between primers and target DNA. In this experiment, the number of mismatches is increased from 0 to 2. An amplification will be performed again. Now, amplicons are obtained for both genomes, and they are also visible in the electrophoresis. In cases where amplification is obtained for both E. coli genomes, a question related to stringency and a link to jump to step number 6 are shown. In step 6, if the amplicons obtained in the PCR reaction are different, that is to say, the DNA sequences of the amplicons are different, the service will let the user know, and design of a PCR-RFLP experiment will be possible. In cases where the sequences of amplicons are identical, a PCR-RFLP experiment cannot be performed, and a new selection of primers for PCR is required in step 3. To complete step 6, restriction endoneucleases for the PCR-RFLP experiment must be selected. A tool to facilitate this selection has been integrated in the exercise. The tool will test the ability of commercial endoneucleases to digest the PCR amplicons obtained in the previous step, and it will show only those endoneucleases which cut the bands from one strain or the other differently. Selection of endoneucleases may be restricted based on the number of bases recognized by the enzyme. Available values are 4 to 8, or based on type of endonuclease. The ones yielding blunt ends, the ones yielding overhand ends, or all of them. Endonucleases that recognize degenerated nucleotides may be removed from the results. After selection, the list of endonucleases is requested. The results page shows the alignment of both amplicons at the top. In this example, the sequences show 4 positions, where nucleotides differ between the amplicon from E. coli K12 and the amplicon from the uropathogenic strain. The different cleavage patterns that restriction endonucleases produce when cutting the sequences will be due to these differences. Below the alignment, a table with the list of restriction endonucleases is shown. Let's look closely at one of those rows. In each row, the restriction enzyme and a representation of the recognition sequence and the cleaved pattern are shown. In this case, enzyme ACO1 will cleave the amplicon obtained from E. coli K12 in position 100. The amplicon, the uropathogenic strain, is not cleaved. Often, 2 or more commercially available endonucleases with identical recognition sequences and cleavage patterns are available. The enzymes with these characteristics are called isoschismers, as, for example, in the noted lists of enzymes. The selection of the exact enzymes to use in wet experiment depends on factors like the temperature of the reaction, the buffer, and so on. Clicking on a small image next to each enzyme will show the commercial source of each endonuclease. We will select one of the available enzymes, or groups of enzymes, to continue. In this example, we will choose the endonuclease ACO1. The service will show the selection, and clicking the next link will allow us to jump to step 7. In this step, a PCR-RFLP experiment is simulated. The in silico experiment requires that we simulate the PCR reaction again, and in a second step, digest the amplicons obtained in the PCR. As in step 5, in this example, the number of mismatches allowed between primers and target DNA must be increased to decrease the stringency of the PCR reaction so that amplification is obtained for both genomes, and then the experiment is performed. The service shows the PCR results first. As expected, amplification is obtained for both strains of E. coli, and restriction results are shown. As the amplicon from E. coli K12 is cleaved, 2 DNA fragments are obtained. The amplicon from the uropathogenic strain is not cleaved, and a unique fragment is generated. Band pattern simulations are also provided, so that the different patterns are easily distinguishable in the image. It must be pointed out that, when very small bands are obtained by digestion, they will run out from the gel. At the very bottom of the results page, a question related to the latter idea is formulated. If the PCR-RFLP experiment allows us to distinguishing both strains, clicking the next link will allow us to jump to the final step. Step 8 includes a summary of all the information of the previous steps. At this point the exercise is finished. 2 links at the bottom of the page allow the user to extend the PCR and PCR-RFLP experiments to additional, sequenced E. coli strains. By clicking the first link, agrosgel simulation of all PCR amplified bands is shown. In this example, amplification was obtained for all E. coli strains. To identify each sample, the user must place the cursor over the sample number, and the identity of the genome and length of bands will be shown. By clicking on the length, the sequence of the fragment is retrieved. In the lower part of the page, a button allows the user to align and compare all amplicons. We will click the button to show a typical result. Before the alignment results, all the fragments are shown as FASTA. And alignment result from cluster W is provided. A form located at the bottom allows the user to compare and group the sequences. 2 methods are available to compute distances: an uncorrected one, and the Jukes Cantor. A gap penalty value may be set up. To compute the distances, the button must be clicked. In the upper part of the page, the amount of different sequences are shown. In this example, the 29 amplicons, each one from one E. coli genome, are grouped to yield 14 different sequences, from A to N. The groups are identified in the text area. For example, 5 amplicons are grouped as type A sequences, 4 amplicons as type B, and so on. On the same page, an image shows how the sequences are clustered by using the neighbor joining method, and next to the image, the clustering is shown as text. Additionally, computed distances may be retrieved. It's worth pointing out that the clustering patterns may group the genomes very differently when the experiment is performed with amplicons from different genes. Consequently, comparing results from different experiments allows us to understand the weaknesses of the methods that establish relationships among genomes by searching only one short sequence per genome. Back in Step 8, a PCR-RFLP experiment may be simulated with all complete E. coli genomes. In this case, 1 or 2 bands are obtained from each sample. 1 band when the amplicon is not cleaved, and 2 when the band is restricted. The discriminatory power of an experiment that searches PCR-RFLP results with a unique restriction in nucleus may be poor for epidemiology in cases like the one in the example, with only 2 patterns. But the combination of data obtained with different enzymes may improve the results. We hope you find this online exercise helpful for your teaching. If you are planning to use this service with over 10 users simultaneously connected to our server, please let us know in advance. A contact form is available on the main page of the service.